Even 30 meters under the rough waters and over sandy bottoms, I've heard of something called climate change and ocean acidification. I feel vulnerable along my 80 centimeters. My name is Dicentrarchus labrax, but I'm also known as European sea bass. My species can be found in eastern Atlantic, but also in the Black and the Mediterranean seas. You can find me in coastal water in summer, but when the weather is colder, I prefer deep water. I spend my days hunting crustaceans, fish, and even cephalopods, but I feel like the acidification of the ocean has an impact on the ecosystems where I live. I've heard that it can lead to a risk for my health and the development of my progeny. I'm interacting with microorganisms in my aquatic environments, and bacteria are a huge part of my microbiota. They have an important role for the maintenance of my good health, and it modulates many physiological activities like the neuronal signaling, behavioral control, immune response, and energy metabolism. Do you know if climate change and ocean acidification have an impact on the composition and structure of my microbiota? Best regards, Seabass.
Hello, I'm Océane Reneteaud. I'm a first-year student of the MARRES program. My immersion project is called Microbar. I'm working on it at l'EMAR, a joint unit of Ifremer, with Dominique, Gwenaël, and Cyril. I also receive advice from Solène. The goal of the Microbar project is to understand the impact of climate change on European sea bass. We want to determine the composition and structure of the bacterial communities that we can find on its skin, gills, and gut. According to the RCP 2.6 scenarios for 2100, sea bass juveniles have been acclimatized to different pH condition and temperature condition. At the end of January, they have been sacrificed in order to get samples from the gut, the gills, and the skin. My task during the internship was to make DNA extraction, prepare libraries, and also analyze the data. Before this, I had to determine which was the best DNA extraction protocol for the amplification of the bacterial 16S RNA, which used the hypervariable V3-V4 region and the primer 341 and 785. Other research projects had to sacrifice sea bass. In order to have supplementary material to try my different protocols, I've been lucky enough to dissect with them and to get the intestine of the fish. I tried 3 different protocols on these fish samples and also on the food fish samples. So I tried the CTAB with a classical phenylchloroform extraction. I also tried 2 commercial kits, fast DNA spin kit for soil and DNZ pyrolyzer for soil. The first step is to perform a lysis on the tissue to give access to the bacteria associated with the tissues. To quantify the quantity of the extracted DNA, I did qubit analysis and epoch analysis. I also did classic PCR with gel electrophoresis. So the results show that CTAB gives no DNA, but only fish RNA. And for the best DNA amplification, it was with the kit Fast DNA Spin Kit for soil. I followed a metabarcoding and bioinformatic formation. Metabarcoding is the study of the taxonomic diversity of the structure of microbial communities between 2 samples with a particular DNA fragment and specific sequences. I also discovered how to analyze data with Samba workflow. It analyzes environmental DNA data to assess the diversity of microorganisms contained in a sample and allows for standardization and comparability of environmental DNA metabarcoding analysis. During the coming weeks, I will perform DNA extraction with a chosen protocol on the fish acclimatized to acidification of the ocean. This project is very complete, but it's only the beginning. That's why you should join us to answer to the question if there is an impact of the bacterial communities on European sea bass.